what are the advantages for the organization of DNA into a chromosome? The packaging of a DNA into a chromosome is very important because a chromosome is going to the compacted form of the DNA. To fit into the nucleus or a cell, even a cell is going to be lesser in diameter than the DNA, because DNA is going to be 2-meter length, but cell is going to be lesser in length than the DNA. Therefore, the compaction of a DNA is very important to fit into the cell and into the nucleus. Second is going to be packaging of a DNA. Packaging of DNA is very important because it is a packaging of DNA into a chromosome is what makes a DNA to be inert and inactive. The inactive form of DNA is very important for the stability of a DNA into the chromosomes. Therefore, it protects the DNA from the DNA damage. The DNA damage is not taken place because of the inactive form of DNA. If the DNA is naked without compaction, it can become unstable inside the cell. Therefore, the chromosomal DNA is found to be very stable form. Next, the stability of a DNA is very important. Why? It's a main question. The stability of a DNA is very important because the DNA stability causes it to be inactive form and stable form. When the DNA is stable, it can be used for next cell division to make a daughter DNA and to inherit the characteristics. Therefore, a packaging of a DNA to transmit into the daughter cells is very important. So that the stability is important. The stability can be made by chromosomes. For the next set of daughter cells, for each set of daughter cells, the stability is very important, which can be helpful for the cell division. Otherwise, when it is unstable, it gets disintegrated. When it gets disintegrated, the DNA gets disintegrated, it leads to loss of certain characteristics. But when it is stable, it can be, there is no disintegration, no loss of characteristics. Therefore, cell division occurs and the characteristics of